Bismillah ar-Rahman ar-Rahim. My name is Dr. Khalid Abul Bashar. I would like to welcome you all to my YouTube channel. Today is my second lecture. It is about cell cycle part 1. I will be talking about the phases and regulation of the cell cycle. The last lecture was about Mendelian genetics, and before going to any further in Mendelian genetics, and before talking about linkage and crossing over, I think it is very important to shed some light on the cell cycle and different phases of the cell cycle and different processes that take place in the cell cycle to understand crossing over and recombination and linkage properly. So this will be the part 1 of the cell cycle, because next lecture will be about also a continuation of the cell cycle. It will be about mitotic and mitosis and meiosis, mitotic and meiotic division, cell theory. Talking about cell cycle, I think we need to speak about a little bit about cell theory. Cell theory states that living organisms are made up of cells, and the cell is the basic unit of structure and organization in organisms, and all cells arise from pre-existing cells. What actually concerns us in this lecture is the last sentence, that is, cells arise from pre-existing cells. If we take human being as example, we see that a trillion and trillions of cells from which human being is made are actually a product of a cellular division of a single cell, that is a zygote. So the zygote divides again and again and again in order to produce a complete human being. So a cell division is a very important process for the survival, differentiation, and the growth of multicellular organisms. Cell division is not the cell cycle. Cell division is part of the cell cycle. So just to avoid confusion, that to think about cell division as the cell cycle, we need to understand that cell cycle is divided into phases. One of these phases is cell division. So what is a cell cycle then? Cell cycle is a series of events that are tightly controlled, and they lead to a cell division and duplication of a cell. A series of events lead to cellular division and duplication, and they are under a tight control of a number of proteins called cyclins, cyclin-dependent kinase, and cyclin-dependent kinase inhibitors. These series of events actually divided into a number of phases. So the series of events divided in, we can divide it either into 2 phases or 4 phases. We can say that the main goal of cell cycle is to duplicate, to produce 2 genetically identical daughter cells, and the actual cell division is the final point of the cell cycle is the actual cell division. So the actual cell division is called the mitosis. This mitotic phase, or the mitotic phase, is the final step or the final phase in the cell cycle, in which cells are actually divided into 2 daughter cells. This mitotic phase is followed by a phase called G1 phase, and the G1 phase is again followed by another phase called S phase, in which the DNA is replicated, is synthesized. So it is called the synthesis phase or the S phase. The S phase is followed by the G2 or the second gap or gap2 phase. So we can look at the cell cycle from uh, two different perspectives. That is, a cell cycle consists of two phases: mitotic phase, M phase, and all these three phases are collectively known as interphase. So you can say that the cell cycle is divided into two phases: mitotic phase and the interphase, or we can say that the cell cycle is divided into four phases: M phase, G1, S phase, and G2 phases. So either way, it is correct. A cell cycle is a two-phases cycle or a four-phases cycle: M phase and interphase, or M phase, G1, S, and G2. Uh, uh, um, before and after the M phase, before and after the cellular division, the M phase, the mitotic phase, there is a resting phase. We cannot say a resting in the sense of the cell do not do anything, but there is nothing like cell division, actual cell division. This phase, the phase that follow the M phase is called the G1 phase. It is directly or immediately follows a cell division. It is also called a pre uh, or post-mitotic pre-synthetic phase. Post-mitotic and pre-synthetic phase. This is the G1 phase. Let's talk about these phases in details. The G1 phase. It is actually the first phase in the interphase, and G phase immediately follow the cytokinesis. Cytokinesis is the actual cell division when the cell divides into two. This process is called cytokinesis. We'll talk about it when we talk about mitosis. So the phase that immediately follows the actual cell division is called the G phase. As I said, it is also called a post-mitotic pre-synthetic phase. Pre-synthetic phase. So it is a post-mitotic pre-synthetic phase, or it is a gap phase, and it follows the cell division. So what happens during this phase? What happens to the cell? After cell division, a cell division actually a product of a single cell dividing into two, giving two daughter cells. Cell division produces from one single cell produces two daughter cells. So understandably, these produced daughter cells are smaller in size and less in the cellular content. So the fate of these cells, these newly produced cells, actually, if it follows all the steps in the cell cycle, they will be prepared for next cellular division. So these cells, which are produced from a previous cellular division, they are smaller in size and less in the cellular content, so they need to grow in size, to increase in size. So what happens in the G1 phase, actually the cell increases in size and increases its cellular content. So after the cell gets divided in the M phase, once they enter the G1 phase, cells start increase in size, grow in size, and increase their cellular content, preparing the cell for upcoming cellular division. Also in the G1 phase, the cell start accumulating all the required materials for the coming phase, which is the S phase. So the cell will start uh, making proteins which are necessary for the S phase, and they make messenger RNA and accumulate all the nutrients during this period, which are very important to have during the S phase. So all the enzymes and proteins and biomolecules required during the S phase, they will be synthesized and accumulated during the G1 phase. And the cell is not allowed to progress from the G1 phase to the S phase unless it is well prepared and everything is in order. After ensuring all this, then the cell is allowed to be processed to proceed. The cell is allowed to proceed from the G1 to the S phase. To progress from G1 to S phase, there are proteins needed for that. We have cyclin proteins, cyclin D and cyclin E, and then we have cyclin dependent kinases, cyclin dependent kinase 4, cyclin dependent kinase 2, cyclin dependent kinase 6 are very important for the cell to progress, to proceed from G1 to S phase. I will talk in details about the role of cyclin and cyclin dependent kinase in the control or regulation of the progression of the cell from one phase to another. Cell in the G1 phase normally takes the longest time of the cell cycle. So the longest time of the cell cycle normally in the G1 phase. Sometimes the cell uh, 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 leaves cell cycle from the G1 instead of going to S phase. They leave the cell cycle through differentiation. So if the cell got differentiated, it is said, and it stops any further division, so it is said to be in the G0 phase. So we have another phase that is called the G0 phase, and the G0 phase is said to be a state of quiescence, not like cell senescence. The G0 phase, actually the cell is out of the cell cycle, but there is a chance that these cells which are present in the G0 phase, they have the chance to go back and re-enter the cell cycle. Cells in the G0 phase are either differentiated cells or cells that are lacking an extracellular or intracellular signals which are important to which are important for the cell to proceed from the G1 to the S phase. So for the cell to proceed from the G1 to S phase, it also requires an extracellular signals and intracellular signals to show that the environment outside the cell and inside the cell are appropriate for the cell to go to the next phase. If the environment, extracellular environment and intracellular environments are not suitable for the cell to proceed in the cell cycle, then the cell will just leave the cell cycle and stay in a G0 phase, which a quiescence cell, a quiescence state. As I said, it is not a similar state as a senescence state, because the senescence state, the cell cannot re-enter the cell cycle. Instead, it is in a state of physiological aging, and it will go directly to cell death. But the quiescent cell or the cell in the G0 and the G0 phase, they can enter if the condition shifted from the unfavorable to the favorable condition that that is suitable for the cell to proceed to the S phase. Now we are here. We are in the G1 phase. We said that after the cell divides, each daughter cell is said to be in the G1 phase, and the G1 phase, the cell is actually small in size. They start growing in size, increases increases its cellular content. All the mitochondria and Golgi apparatus and all the cytoplasmic materials are actually increased during the G1 phase. And for the cell to proceed from the G1 phase to the S phase, there are a number of proteins required for the cell to go from G1 to S, and the cell cannot be allowed to proceed to the S phase unless all the materials and molecules required for the S phase are completely ready, and the cell have all the internal and external requirement for the cell to proceed. If the condition externally and internally are not suitable for the cell to proceed on, they will leave cells will leave the cell cycle and will remain in a state of quiescence, and this is called the G0 state. If the cell in the G1 phase cell in the G1 phase, if it is allowed to move on to the S phase, in the S phase it is a synthesis phase. In the S phase, the peculiarity of the S phase, that is DNA replication. This is a phase in which the DNA is replicated and all the genetic material, the chromosomes are duplicated so that the cell prepare itself for a cellular division, so that when the cell reaches the M phase and when the cell divides into two, there is enough material to be divided in the nuclei of both daughter cells. So the DNA is replicated in the S phase, and S phase is a very important for the cell to enter the cell division. It actually takes around 7 hours on average for the cell to replicate its DNA, and during this phase also centrosomes are duplicated. It's not only the DNA that is replicated, but also centrosomes are duplicated, and there are number of proteins which are very important for the cell to enter the S phase. These are cyclin A, CdK2 complex, which plays a very important role in initiation of DNA replication. So for the cell to start the S phase, it requires the presence and activation of the cyclin A CdK2 complex, and also the centrosome duplication requires cyclin E CdK2 complex. So we have a number of protein, number of cyclin dependent kinases that are needed for the cell to proceed from G1 to S phase, and we have another type of proteins and cyclin dependent kinases that are required for the initiation of DNA replication and that are required for the duplication of centrosomes. So it's very important for these proteins and these proteins and these enzymes to be present for the cell to enter and proceed with its S phase processes. So the S phase, during this S phase, as I said, the DNA will be replicated. The S phase is followed by a G2 phase. The G2 phase is a pre-mitotic post-synthetic phase. It's a pre-mitotic post-synthetic phase. So it is a phase that is precede the mitosis and follows the synthesis S phase. During this phase, the cell basically prepare itself for the cellular division. How? It synthesize all the proteins and messenger RNA, and it accumulates all the nutrients required during the M phase, and it also loosens up the direct contact to neighboring cells so that it prepares itself for the actual cell division. It takes the round shape and increases in size. So all these events happens and occurs during the G1 phase. The DNA fidelity check point also ensure a complete replication. So the cell is not allowed to go from the G2 to the M phase unless the cell ensures that its DNA is completely replicated and there are no major errors in the DNA products, and then the cell also, as I said, they synthesize all the proteins and the messenger RNA needed for the mitosis. Again, we have a number of proteins that are very important players for the progression of the cell from the G2 phase to the M phase. This is a cyclin B CdK1 complex. It's a very important complex to the cell to proceed for the M phase. This phase is very short. It actually takes from 2 to 4 hours in time. So now we talked about the three phases of the interphase: the G1 phase, the S phase, and the G2 phase. I will not be talking about the M phase, because the M phase I will talk about it in a completely separate lecture, next lecture maybe. I will talk about mitosis and meiosis. Now I will go directly to the regulation of regulation of a cell cycle is carried out by a number of proteins. We have three groups of proteins that are very important players in the regulation of the cell cycles. These groups of proteins are cyclins, cyclin-dependent kinases, and cyclin-dependent kinases inhibitors. Cyclin and cyclin-dependent kinases, these are a positive regulator, while cyclin-dependent kinase inhibitors is a negative regulator. I will talk about this in details. The action of cyclin and cyclin-dependent kinases is actually they turn specific proteins, especially enzymes and transcription factors, on and off by phosphorylation. So what they do, the cyclin and cyclin-dependent kinases, they actually turn on and off a number of proteins, very important proteins in the cell during different phases of the cell cycle, on and off by phosphorylation. Events in every phase of the cell cycle are carried out by a specific number of proteins, specific types, specific proteins, specific enzymes and transcription factors. These proteins, which are very important for different phases in cell cycle, they must be synthesized. These proteins must be synthesized, activated, and inactivated at the right time. So how can we synthesize, activate, and inactivate different enzymes, different proteins needed for different phases in the cell cycles? Cell cycle is regulated by sequential formation, activation, and inactivation of cell cycle regulatory molecules. So what happens? A cell in one of the phases requires number of proteins, number of enzymes, number of molecules. So by activation, formation first, then activation, then inactivation of certain regulatory proteins, these proteins will ensure the production and activation of the important proteins and enzymes required for that phase, that's cell cycle phase. So through activation, sequential activation, formation first of cyclin and cyclin-dependent kinases complex, or cyclin dependent kinase inhibitor, and then activation after formation or inactivation, we ensure that the specific and the right combination of proteins are present in a specific cell cycle phase. So let's talk about cyclin, for example. Cyclins cyclins are number of proteins that specifically bind to a cyclin-dependent kinases. And cyclin-dependent kinases and cyclin, when they bound together, they form a complex. This complex has the activity, the catalytic activity of transferring the phosphate group from ATP to a downstream protein, to another protein. So cyclin and cyclin dependent kinase normally bind together and form a complex. This complex has the catalytic activity, the kinase activity, which is transferring the phosphate group from ATP to another molecule. So at specific phase of the cell cycle, these cyclins and cyclin dependent kinases form. Cyclins are family of proteins that are synthesized and degraded during each cell cycle. They are actually synthesized. We have a number of cyclins. Some cyclins are produced during, for example, G1, and by the end of the G1 phase, these cyclins are degraded. And when the cell enters S phase, another type of cyclins are produced, which are required during that phase. And by the end of that phase, the cyclins will be degraded. And then when the cell enters the G2 phase, then another type of cyclins are produced, and by the end of the G2 phase, the cyclin will be degraded. So they are subjected to a continuous synthesis and degradation cycles. These cyclins, they all the cyclins, there are eight cyclins known. There are 8 type of cyclins. They all share a 150 amino acid region of homology. This region is called cyclin box. The cyclin box is a site where the cyclin actually binds to an end terminal of a cyclin dependent kinase. So the cyclin and cyclin dependent kinase binds through the cyclin box, which is a region in the cyclin that is specific for the binding of cyclin to cyclin dependent kinase. The function of cyclin in this complex, in the complex of cyclin dependent kinase, is the activation of cyclin dependent kinase first. So the cyclin will activate cyclin dependent kinase. Another thing that is a cyclin targets the complex to the specific substrate proteins. So there are 2 functions for the cyclin. First, cyclin will bind to cyclin dependent kinase and activate the complex, activate the cyclin dependent kinase, and then the cyclin will target the complex, the whole thing, to a specific target substrate protein. And the other part of the complex, which is the cyclin dependent kinase, has a function, has a catalytic function, the kinase function. So it is the enzymatic part which has the catalytic function of transferring the phosphate group to other proteins. So by forming a complex with a cyclin, now the cyclin dependent kinase is active and it can carry on its catalytic activity. When each subunit, cyclin and cyclin dependent kinase, are separated, they have no catalytic activity. So the formation of cyclin dependent kinase complexes activate the cyclin dependent kinase enzymatic activity, and the cyclins will target the whole complex to the specific proteins. As I said, cyclins are the level of cyclin is normally fluctuating during the cell cycles. At the beginning of certain cell cycle phase, we find the level of cyclin, certain cyclin very high, and by the end of that phase, the level will just drop down, and cyclin will disappear, and then in the next phase another type of cyclin will increases, the level of it will increase, and then by the end of so it is in a continuous fluctuation, the level of cyclin. But on the other hand, the cyclin dependent kinase, the level is relatively constant throughout the cell cycle. The cyclin-dependent kinases are actually serine threonine protein kinases, and we have 9 of them unknown by today. We have number of cyclin-dependent kinases unknown. We have 8 cyclins and 9 cyclin-dependent kinases are known, and they are, as I said, serine threonine kinase type of proteins. They turn specific proteins on and off by phosphorylation. So the kinase activity is transferring the phosphate group, phosphorylation of certain proteins to turn them on and off. So now in this table we have a name of a cyclin dependent kinase complex. We have the name of the complex and the name of cyclin subunit and the name of cyclin dependent kinase subunit. So binding of cyclin D with cyclin dependent kinase 4 and cyclin D with cyclin dependent kinase 6 will form a complex known as G1 cyclin dependent kinase. So this type of complex is present in the G1 phase. So this complex, which is made of cyclin D and cyclin dependent kinase 4 and CDK6, when they bind together, CD4, CDK4 and cyclin D, or CD6 and cyclin D, they make what is known as a G1 CDK. So this complex is normally found and function during the G1 phase, and it is made of cyclin D and cyclin dependent kinase 4 and 6. We have another complex that is called the G1S CDK. It's a complex that activates the transition of the cell from G1 to S. It is called G1S CDK, and it is made up of cyclin E and cyclin dependent kinase 2. We have another complex made up of cyclin A, a complex of cyclin A and CDK2, which is found in the S phase and function in the S phase during the the DNA replication, and it is called the S-cdk. And finally, we have another complex called M-cdk, which is a very important cyclin dependent kinase complex that is function during the M phase, and it is made up of cyclin B and CDK1. So this is what is to be said about cyclin and cyclin dependent kinases. To wrap things up, these are very important regulatory proteins. They are found in the different phases of the cell cycles, and cyclins and cyclin dependent kinases normally they bind to each other forming complexes, and these complexes have the catalytic activity of a kinase enzyme, so they transfer, so they phosphorylate number of proteins during the cell cycles, and for the cyclin dependent kinase to carry on the enzymatic activity, it needs to bind to the cyclin, which activates the enzymatic part of the complex, and the cyclin also direct the complex to its specific protein substrate. We have another regulatory proteins, which is a cyclin dependent kinase inhibitor. cyclin-dependent kinase inhibitor. It, it is actually a family of negative regulators of the cell cycle. The inhibitors, this family is comprised of two structurally distinct families. It is made up of two families. One family is called the INK4 or inhibitors of CDK4. This is one type of CDK inhibitors, INK4, and the other one is called CyclKIB. INK4, these are inhibitors of cyclin-dependent kinase 4. This include P14, P15, P16, P18, and P19. P14, 15, 16, 18, and 19, they belong to an INK4 cyclin-dependent kinase inhibitors. They specifically inhibit G1 cyclin CDK. All these P4, P14, B15, B18, B19, and B16, they inhibit this complex. And how they do that? They bind to the complex. After the formation of the complex, they bind to the complex and prevent its phosphorylation activity. So the INK4 inhibitor, they all bind to the G1 CDK because it is made up of CDK4, so they are called INK or inhibitor of CDK. So after the formation of this G1 CDK, the inhibitor will bind to this complex and preventing its phosphorylation activity. Another type of inhibitors, CDKs inhibitors, they are called CyclKIB or CDK interacting proteins, kinase inhibitor proteins. CyP, CIP, and KIB, KIP. It is CyclKIB, it is CDK interacting proteins, kinase inhibitor proteins. These are number of proteins. They include P21, P27, and they display a broader inhibitory effect. Instead of working on one complex, they work on number of complexes, cyclin-dependent kinase complexes, and they affect they function throughout the cell cycle. In all the phases of the cell cycles, we can find CyclKIB inhibitors, and they can inhibit cyclin E and G1 cyclin-dependent kinase CDK and cyclin D CDK6, cyclin A CDK2. So number of complexes this CyclKIB can inhibit during the phases of the cell cycle. So now these are the proteins, these are the molecules involved in the regulation of the cell cycles. First, cyclin-dependent kinases, and finally cyclin-dependent kinases inhibitors. Now we go to the regulation pathway we can talk about that in just generally, and after that we go to specific details of a regulation pathway of a cell cycle. So the first point in the cell cycle, what happens is that activation of G1 CDK. How it happens? First, the synthesis of cyclin D takes place in the cycle, in the cell. Synthesis of cyclin D, and this cyclin D will bind to a CDK4. Cyclin D will bind. I will show you how cyclin D is actually synthesized and activated. Cyclin D is synthesized in the cell. This is the first step in the cell cycle. Cyclin D is synthesized, and then it goes and bind with a CDK4, forming a G1 CDK. This is a complex made up of cyclin D and cyclin dependent kinase 4, as we have seen here. G1 CDK, it's made up of cyclin D and CDK4. So CDK4 and cyclin D will make a G1 CDK. So now this is an active complex, a kinase complex. This will turn on a synthesis of number of proteins. Number of protein. All this happen in the early stage of phase 1 of G1 phase. G1 phase. So in the G1 phase, the very early stage in the G1 phase, cyclin D bind to cyclin dependent kinase 4, forming a G1 CDK, and this G1 CDK will turn on the synthesis of cyclin E. How? Cyclin E and a number of other proteins. This is actually activates transcription factors that are responsible for the synthesis of cyclin E. So this will activate this complex will activate transcription factor, specific transcription factor for the synthesis of cyclin E. Now the cell in the G1 phase, after the formation of this complex, started to activate the transcription factor responsible for the synthesis for the transcription of cyclin E gene. So the cell will synthesize the cyclin E due to the activation with this complex. Cell will synthesize the cyclin E, and now cyclin E is present in the cell. This cyclin E will bind to cyclin dependent kinase 2. Cyclin E will bind to cyclin dependent kinase 2 to form a G1 S-cdk. To form a G1 S-cdk, when this increases to a certain limit, when this complex increases to a certain limit, it triggers the transition of the cell from late G1 to S phase. So now the cell first synthesizes cyclin D. Cyclin D will bind to CDK4, forming a complex called G1 CDK. This complex will activate the transcription factor of a gene. Which gene? Number of genes. One of them is cyclin E gene, and this will turn on activation of the transcription factor, means turning on the synthesis of cyclin E. So the cyclin E will be produced, and when the cell produced enough amount of cyclin E, this cyclin E will bind to CDK2, forming a complex called G1 S-cdk, and this complex will triggers when it is in enough amount. It will trigger the transition of the cell from G1 to S phase. So now the cell is allowed to go to the S phase by the action of G1 S-cdk. Now the cell moved from the G1 phase to the S phase. What happens in the S phase? In the S phase, the cell will start making cyclin A. When cell enters the S phase, it makes cyclin A, which binds to CDK2. Binding of cyclin A to CDK2 will form an S-cdk. This is a complex. This complex, in turn, it will transcribe, or it will trigger or activate the transcription factor for number of enzymes and protein required for the DNA synthesis. So this is very important for the DNA synthesis. So now in the S phase, production of cyclin A will bind with the CDK2. Cyclin A will bind to the CDK2, forming an S-cdk. S-cdk, this complex will activate a number of transcription factors so that a number of enzymes will be produced These enzymes are very important for the synthesis of DNA. Now the DNA will be synthesized because all the enzymes required for the synthesis of DNA are there and they are produced. So the DNA will be synthesized. Now we are in the S phase. Okay, after that, in the S phase itself and G phase, in the S phase, and continue and this in the S phase and in the G phase, cyclin B will be produced. So cyclin B will be produced later in the S phase and early in the G2 phase, G2, S and G2 phase. Cyclin B will be produced. Cyclin B will then binds to a CdK1 to form an MCDK complex. Now formation of this complex, what will happen when MCDK reach a threshold activity, when it is there is an enough amount of CDK MCDK complex, it will trigger the transition of the cell from G2 to M phase. Now the cell will be allowed to go to M phase. So this is in general the regulatory pathway of the cell cycle. Production of cyclin D, binding of cyclin D to form a G1 CDK. This will activate the synthesis of cyclin E. Production of cyclin E produced will bind to CDK2, forming a G1 S-cdk. This will activate the transition of a cell from G1 to S, and then in the end, in the S phase, cyclin A will be produced, and this cyclin A will bind to CDK2 to form an S-cdk. The S-cdk in turn will activate number of enzymes that are very important for the DNA synthesis. Also in the S phase, late in the S phase and early in G2 phase, cyclin B will be produced, and this cyclin B will bind to CDK1, forming an MCDK. This MCDK will allow the cell to be will allow the progress of the cell from G2 to M phase. Now we go in details to regulation of cell cycle. In the G1 phase, regulation of G1 phase, that is the formation of G1 CDK, which will open or trigger the G1 S-cdk. So the formation G1 phase, the regulation of G1 phase, actually through the formation of G1 CDK complex, and this will trigger the formation of G1 S-cdk. So let's talk about it. Let's talk about it in details. G1 phase is the only time in the cell cycle where cells are sensitive to signal from their external environment. So in G1, the only time that is a cell is sensitive to extracellular signals, growth factors and mitogens that can affect the cell cycle and stimulate the cell to enter the cycle. This can only happen during the G1 phase. So the G1 is the only time where the cells are sensitive to extracellular signals. So cell require growth factor in the G1 phase up to a certain point. So without growth factor from extracellular environment, cell cannot proceed in the G1 phase. But if we go and provide the cell with an extracellular growth factors, these growth factors will stimulate the cell to go on in the cell cycle until a certain point in G1 phase. After that point, the cell will be independent of a growth factor, a stimulation of the growth factor. So in order to move from early G1, from the time where the cell is a growth factor dependent cell to a late G1 phase where the cell is independent of any stimulation, extracellular stimulation, cell must synthesize cyclin E. So the very important part of this phase is synthesis of cyclin E, because synthesis of cyclin E is a very important step for the cell to move from a state where the cell is totally dependent on extracellular stimulation to a point where the cell is completely independent of any extracellular stimulation. So the transcription of cyclin E, the production of cyclin E, transcription of this gene, cyclin E gene. Transcription of cyclin E gene requires a transcription factor. It is very well known that is the transcription of a gene requires a transcription factor. And there are specific transcription factors for every gene. One of the specific transcription factors required for the transcription of cyclin E is E2F. So E2F is a transcription factor for transcription of a cyclin E gene. E2F is normally bound to a cyclin E gene promoter. In the gene, the promoter of this gene, cyclin E gene, is normally bound to an E2F transcription factor. But when the cell is non-dividing, in a non-dividing cell, what happens? This transcription factor is actually inhibited. Since the cell is not dividing, the transcription factor is inhibited by what? By a protein, by protein bound to it. This protein is called the RP protein, or retinoblastoma protein. So the inhibition of E2F transcription factor, so that it does not carry the transcription of cyclin E in a non-dividing cell, is done by the binding of RB protein or retinoblastoma protein. So binding of an RP protein to a transcription factor which is already bound to a cyclin E gene promoter will prevent the transcription of the cyclin E gene. So it will prevent the production of cyclin E. Now what happens? What about the RP protein? What is the RP protein? It is a type of a protein that is activated by underphosphorylation and inactivated by hyperphosphorylation. If we added phosphate group to this RP protein, the protein will turn inactive. And if we remove the phosphate group from that protein, from RB protein, then the protein will be active. So the protein, this RB protein, retinoblastoma protein, is activated by underphosphorylation and inactivated by hyperphosphorylation. Addition of phosphate will inactivate the RP protein. Removal of phosphate will activate the RP protein. So in dividing cells, this is in a non-dividing cells, normally the transcription factor is bound to the promoter, but it is inhibited by binding of an RP protein. Okay, what happens when the cell divides? So in a dividing cell, when the cell divides, we have a G1 CdK. G1 CdK increases, as we said in the previous slide. G1 CdK. This is G1 CdK. Now we are not talking about cyclin D. We are going step before what happens, how cyclin D is produced. We are talking about how cyclin E is produced. Now in a dividing cell, G1 CdK, this complex, which is formed from a CDK4 and a cyclin D combination, this will form a G1 CdK. The formation of this G1 CdK, what will happen? The G1 CdK activity increases. since G1 CDK formation took place, the G1 CDK activity will increase. So, and it will phosphorylate the RB protein. Now G1 CDK increases, and it is a kinase. CDK is complex, and this complex is made up of cyclin D and CDK 4, cyclin D and CDK4. Cyclin D and CDK4 will form a G1 CDK, and now it is an active complex. This active complex will phosphorylate now in a dividing cell. G1 CDK is formed, active complex, it will phosphorylate the RP protein. And we said that the RP protein, when it is phosphorylated, it will become inactive. When it is under phosphorylated, it will become active. So phosphorylation of RP protein means it will become inactive. So inactive RP protein will be released from the E2F transcription factor. It will be released from E2F transcription factor. So now this transcription factor is free. It can go on and do the transcription job. So E2F transcription factor will go on and transcribe DNA polymerases, and it will transcribe cyclin E protein. So it will produce, this transcription factor has many work to do. It will transcribe some DNA polymerases which are required for the S phase, and it will also transcribe the cyclin E gene producing cyclin E. So production of cyclin E, production of cyclin E, this one which will bind to a cyclin D, cyclin dependent kinase 2. Cyclin E then will bind, produced due to the release of RP protein. Why RP protein is released from the transcription factor? Because this complex has phosphorylated the RP protein, and the RP protein became inactive, releasing from the E2F transcription factor. Now the transcription factor is free to move and transcribe the gene. The transcription factor will help in the transcription of DNA polymerase and the transcription of cyclin E. Cyclin E is produced due to the transcription of the gene. Cyclin E is produced. This produced cyclin E will bind to a CDK2, making a complex called G1 S-cdk. G1 S-cdk in turn, at this point when the G1 S-cdk is produced, production of cyclin E and CDK will bind, forming a S-cdk, G1 S-cdk. This represents a transition from a mitogen dependent to mitogen independent cell cycle progression. And at this point, the cell is committed irreversibly to enter the S phase. So at this point, when this complex is produced, is made, the cell is committed to irreversibly enter the S phase. So this is what will happen. Cyclin E transcription is blocked by the RP protein. Why? Because transcription of cyclin E requires E2F cyclin kinase, and cyclin E is not produced because although E2F transcription factor is bound to cyclin E gene promoter, but it is inhibited by the action of the RP protein. When the cell starts to divide in the early phases of G1, in the early stage of G1 phase, the cell will produce a large amount of G1 CDK. This is due to the production of cyclin D and the binding of cyclin D with cyclin dependent kinase 2, making a G1 CDK. This amount of complex, when it increases, its activity will increase. Then it will phosphorylate the RP protein, and we said that RP protein, when it gets phosphorylated, it will become inactive. When it becomes inactive, it will release from the transcription factor, leaving the transcription factor free to carry its job, and the transcription factor then will help in the production or transcription of DNA polymerases, the enzymes that are required for the S-phase, and transcription of cyclin E gene, producing cyclin E, and then cyclin E will bind to a cyclin dependent kinase 2, making or forming a G1 S-cdk. The production of G1 S-cdk is an indication that the cell has moved from an early stage of G1 to a late stage of G1, from a mitogen dependent, from an external signal dependent cell to a completely independent uh, ex external stimulation, completely independent cell. Now we have talked about the cyclin E, but before production of cyclin E, we know that we need a cyclin D. Cyclin D, which is very important for this complex to be made. The complex which helps in the release of the RP protein from the transcription factor. So how can this complex is actually produced? We know that cyclin dependent kinases are normally there in the cells. What govern the formation of these complexes is the production, the synthesis of cyclins, uh, synthesis of cyclins. So if cyclins are there, CDKs are normally found, are normally there in the cell. So the production of cyclin means the formation of the complex. No cyclins, that means no complex. So now we go one step further. Cyclin D regulation through mitogenes. So what are mitogenes? Mitogenes are growth factors. These are substances which stimulate the cell to start the cell cycle. So these are mitogenes, normally called growth factors. For example, in a G0 cell, a G0 cell can enter or re-enter the cell cycle in G1 phase when it is stimulated by a mitogene, extracellular stimulatory molecule, normally protein. When a cell in a G0, it can re-enter the cell cycle again in a G1 phase when it gets stimulated by mitogenes. And these mitogenes are normally are growth factor. They are normally proteins. And cell can be arrested in the G0 when an anti-mitogene is added to the cell. When the mitogene will take the cell from G0 and make it enter the cell cycle in the G1 phase, while anti-mitogene will take the cell from the G1 and make it leave the cell cycle to the G0. So now we have two type of substances: mitogenes, which stimulate the cell from the G0 to enter the G1 and the cell cycle, and the anti-mitogene. These are substances which are responsible of making the cell to leave the cell cycle from the G1 to G0. So one of the mitogenes that are very important in the cell cycle is epidermal growth factor. Epidermal growth factor, or EGF. EGF. What is the EGF, and how EGF normally make the cell re-enter from the G0 to G1, re-enter the cell cycle? Epidermal growth factor is a protein. When it is produced by some cells, they will bind to a surface receptor on the cell. Now we are talking about a cell in a G0 phase, non-dividing. So when a mitogen is produced, EGF, when an EGF is produced by some cells, and this mitogen, this EGF, is released by some cells on the G0 cell. This EGF will bind to an EGF receptor, a surface receptor, on the surface of the EG0 cell, in the in the G0 cells. Excuse me. When the EGF is secreted, it will bind to an extracellular domain of an EGF receptor, which is found on the surface of a cell in the G0 phase. This receptor is normally a surface receptor. It is a protein with a single transmembrane domain. The EGF receptor is a protein with a single transmembrane domain. That is, the protein will pass from the outside external part through the membrane, cellular membrane inside the cytoplasmic. So there is an extracellular domain, we have a transmembrane domain, and we have the cytoplasmic domain. So it extends all the way from outside the cell till the inside of the cell. This is the receptor. We are talking about the receptor. When this EGF, epidermal growth factor, binds to this receptor on the surface of the cell, what will happen? A cytoplasmic domain now inside the cell, the inner part of the receptor, the inner part of the receptor is made up of two domains. It's made up of two units. Now we have a receptor, a long receptor, going from outside the cell to inside the cell. Inside the cell, the receptor is made up of two units, two domains, okay, two dimers, two units. The receptor inside the cell, in the cytoplasmic part, it is made up of two parts, okay. When the protein, this EGF, is bound to a receptor, to extracellular part of the receptor, the inner part, the cytoplasmic part, one of the subunit, one of these parts, I said that inside the cell there are two subunits. One subunit will phosphorylate, transfer the phosphate from ATP to the other part. So one subunit will phosphorylate the other subunit. So this is called a transphosphorylation. So now externally, outside the cell, the EGF is produced by some cells and it is released on the environment, surroundings, and this EGF will bind to a receptor on a non-dividing cell. This EGF receptor to which the EGF will bind has an external or extracellular domain, extracellular part, and intracellular or cytoplasmic part. The cytoplasmic part is made up of two units. When the EGF is bound to extracellular part, one of the two units inside the cell will phosphorylate the other, and this is called transphosphorylation. And the phosphorylation is actually happens in a tyrosine amino acid. That means the transmembrane receptor, one subunit and the other subunit. In the other subunit, the first subunit will take a phosphate from ATP and transfer it to a tyrosine amino acid in the second subunit of the receptor. So the presence of the phosphate on the tyrosine subunit, on the tyrosine residue, on the tyrosine amino acid, give a signal to another protein that is found inside the cell. This phosphorylated subunit in which the tyrosine has been phosphorylated, this will serve as a binding site. Now this will give a signal for another protein to bind to this this subunit. So transphosphorylation means one subunit will phosphorylate the other subunit. After transphosphorylation, we have another protein called GRB2. The GRB2 has a and a domain with SH2 domain. It's called SH2 domain. This SH2 domain recognizes the tyrosine amino acid on which the phosphate is added. So the GRB, through it is at SH2 domain, will bind to the receptor. The receptor. So the GRB first, EGF will bind to the receptor. Then the receptor will transphosphorylate. One subunit will transphosphorylate the other. Transphosphorylation, then followed by binding of a GRB protein to the transphosphorylated subunit. GRB protein, when bound to a transphosphorylated subunit, will then recruit another protein, will come and bind to the GRP, which is already bound to the receptor. So this SOS protein can only bind to a GRB protein which is bound to the receptor. If GRB protein is free, this SOS cannot bind to the GRB protein. So binding of the epidermal growth factor to the receptor will lead to the transphosphorylation. Transphosphorylation will help in the binding of GRB2 protein to the receptor in the cytoplasmic domain, and this bound GRB2 protein will be bound by or another protein. It will recruit an SOS protein. This SOS protein activates GTP binding protein. This SOS protein will activate a protein called RAS protein. RAS protein is a GTP binding protein. RAS protein is a GTP binding protein. This is RAS protein and can be activated by a protein called GAP protein. So GAP protein can activate the RAS protein, and RAS protein is a GTP binding protein. So if there is no GAP protein or the GAP protein is inactive, the RAS protein will bind and activate another protein called RAF protein, which is a protein kinase. So binding of a mitogene, which is epidermal growth factor, will to the receptor, will lead to transphosphorylation of one subunit to another, and then transphosphorylation of one subunit by the other, and then transphosphorylated subunit will be bound by a GRB protein, and this GRB protein will recruit an SOS protein. SOS protein will be activated, and they will bind and activate a RAS protein. Then an activated RAS protein will bind and activate RAF protein, which is a protein kinase. RAF protein, it activates through phosphorylation. It will also transfer a phosphate protein to a protein called MEC protein. MEC protein, in turn, it will bind and activate a protein called MAB protein, or mitogene activated protein kinase. This MAB kinase, this is all called a MAB kinase cascade. We see that the binding of the EGF to the receptor will lead to transphosphorylation. Transphosphorylation will lead to the binding of the GRB2. Then the GRB2 bound GRB2 will recruit an SOS protein. This SOS protein will activate a RAS protein. RAS protein in turn, it will bind and activate a RAF protein kinase. RAF protein kinase will transfer a phosphate to MEG protein, activating it, and this MEG protein will in turn activate a MAB kinase protein, which is a mitogen activated protein kinase. And this MAB, mitogen activated protein kinase, will then activate a cyclin D gene transcription factor. So this will activate a transcription factor for cyclin D gene. So activation of a cyclin D gene transcription factor will lead to the production of cyclin D. Now this is a cyclin D. Cyclin D. Cyclin D, the production of cyclin D requires a mitogen, an extracellular signal. So this can happen through an EGF, epidermal growth factor, will lead to production of cyclin D. Then cyclin D activates, it binds to a cyclin dependent kinase 4, making a complex called G1-CDK. G1-CDK, this is a very important one. We have seen it in the regulation of G1 phase. G1-CDK, when it increases, it releases the APE protein. So the production of G1-CDK follows the synthesis of cyclin dependent kinase, follows the production of cyclin D, and then the production of G1-CDK now will lead to cellular proliferation. So from the beginning, the cell is in the G0 phase. It's not a dividing cell. When EGF, epidermal growth factor, is released and bind to a receptor, EGF receptor, this will lead to a transphosphorylation of the receptor, the cytoplasmic domain. The phosphorylated domain will be the site where the GRB2 protein will bind. The bound GRB protein will recruit an SOS protein. SOS protein in turn will activate a RAS protein, which is a membrane, transmembrane protein, which is a membrane protein, same like G-link binding receptors, G-link proteins, RAS protein, and then RAS protein will in turn bind and activate a RAF protein kinase. RAF protein kinase will activate another protein called MEK, and MEK protein will activate a mitogen activated protein kinase or MAP kinase. This MAP kinase finally will activate a transcription factor for cyclin D gene. This will lead to production of cyclin D, and production of cyclin D will bind to a cyclin dependent kinase 4, forming a G1-CDK, and this will make the cell move from a G0 phase to a G1, and even from G1 early phase to G1 late phase, because the production of CDK is an indicator of that the cell is now moving through a cell proliferation or cell cycle. This is a regulation of cyclin D production through mitogen. We have another pathway through which a cyclin D will be produced. We have another pathway. This pathway is called Wnt pathway, Wnt signaling pathway. Wnt protein, Wnt is a protein that is a mitogen. It is a mitogen, and it regulates proliferation by controlling proteolysis of a key signaling protein. So this protein regulates proliferation, cell division, by preventing or controlling protein degradation or controlling proteolysis of a key signaling protein. Now, Wnt protein prevents a degradation of a certain protein. If that protein is degraded, so the cell will not divide. If the protein is not degraded, then the cell will divide, and the Wnt protein will protect that important protein from getting degraded. So let's see how Wnt pathway goes on. Wnt is a protein, extracellular protein. When it is produced and secreted, it will bind to a surface receptor. It's also a ligand that will bind to a receptor. The receptor is called frizzled. So Wnt protein will bind to a surface receptor called frizzled. So the frizzled, it's a receptor, frizzled receptor is activated when bound to Wnt protein. It will get activated, and it will in turn uh, 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 turn off a protein called GSK3. So the frizzled receptor, after getting activated by binding a Wnt protein, it will turn off a GSK3 protein. A GSK3 protein normally, this protein normally promotes the degradation of another protein called beta-catenin. So normally GSK3 is a protein that promotes the degradation of beta-catenin, a very important protein for the cell cycle. Normally, GSK3 is a protein that promotes the degradation of beta-catenin. But when the Wnt protein binds to surface receptor, the frizzled receptor, this frizzled receptor will inactivate the GSK3. The GSK3 now it's off, so beta-catenin will not be degraded, and it will bind to another protein called TCF. The binding of beta-catenin and TCF will form a cyclin D gene transcription factor. So both TCF and beta-catenin, when they bind together, they form a transcription factor for cyclin D gene. So when this transcription factor is formed, it will transcribe the cyclin D gene, and making a cyclin D, this will trigger the cell proliferation. Now, if Wnt protein is not bound to the surface receptor frizzle, the GSK3 will the GSK3 will not be turned off, and it will always be on, and this will degrade the beta-catenin with the help of a protein called APC. So this APC will hold the beta-catenin so that the GSK3 to a phosphorylated, and the phosphorylation of beta-catenin will lead to the ubiquitination of the protein, the beta-catenin, and later on to the degradation of beta-catenin. When the beta-catenin is degraded, so no cyclin D will be produced. Again, Wnt protein, if produced, it will bind to a frizzled surface receptor, and this frizzled surface receptor will inactivate the GSK3. The GSK3, if is inactivated, then it cannot degrade the beta-catenin, and beta-catenin is a very important protein. When it is there and when it is bound to TCF protein, it will form a cyclin D gene transcription factor. Cyclin D gene transcription factor will then transcribe the cyclin D gene and produce cyclin D, and the production of cyclin D means cellular proliferation. But if Wnt protein is not bound to a surface receptor, it's not bound to the frizzled, then the GSK3 will not be inactivated, it will be active, and with the help of ABC protein, which is adenomatoous polyposis coli, this APC protein, which is a very important protein, which holds the beta-catenin so that the GSK3 will phosphorylate the beta-catenin with the help of ABC, if this is active, when this protein is not there, and this receptor is not activated, so this will be active, so it will not be turned off, it will be turned on, because it's not activated, it's not inactivated by the receptor, so it will be on, and with the help of ABC, both proteins will phosphorylate beta-catenin. Phosphorylation of beta-catenin means a later degradation of beta-catenin, and degradation of beta-catenin means there will be no transcription factor for cyclin D, so cyclin D will not be produced. So this is another pathway through which cell cycle can be controlled. We have another pathway which is an anti-mitogenic pathway. The Wnt pathway and the epidermal growth factor pathway, both are mitogenic pathway. Now we have an example of anti-mitogenic pathway, which is called TGF beta SMAD pathway, SMADs. So this is a transforming growth factor beta, and SMAD proteins pathway. It's an anti-mitogenic pathway. Anti-mitogenic means that the cell will be moved from G1 to G0. Now we are talking about how the cell can be arrested in the G0 phase. So in this anti-mitogenic pathway, the transforming growth factor beta is an external protein. This is a protein, TGF beta, which binds to a TGF beta receptor on the surface of a cell. This receptor is different from the mitogenic receptors, which are tyrosine kinase, but these are a serine threonine kinase receptor. This is a serine threonine kinase receptor. So this is different from the mitogenic receptor, TGF, EGF. So when the TGF beta is bound to its receptor, the receptor will phosphorylate a cytoplasmic protein. This protein is found in the cytoplasm will be phosphorylated by the receptor. By the cytoplasmic domain of the receptor will phosphorylate the SMADs proteins, group of proteins called SMADs. Those proteins, after phosphorylation, they will get activated and they will enter the nucleus. Entering the nucleus, these proteins, SMAD proteins, activated by the phosphorylation by the receptor, TGF beta receptor, they will enter the nucleus. Inside the nucleus, they will transcribe a gene. Which gene? P21. And we know that P21 is one of the cyclin-dependent kinase inhibitors. We have talked about cyclin-dependent kinase inhibitors. It will inhibit a cyclin-dependent kinase. So P21 gene now is transcribed. This will lead to production of P21, which is an inhibitor of cyclin-dependent kinase. So P21 produced will inhibit the CDK2, cyclin-dependent E and A. So this is where it will work. Here, cyclin-dependent cyclin E with CDK2. The formation of this, the movement of the cell from G1 to S will be blocked. Why? Because this complex is inhibited by B21. B21 will inhibit the cyclin E with CDK2. So this will be inhibited. The movement of the cell from G1 to S will be inhibited, and the cell will be arrested in a G0 phase. So this is, in short, the regulation of a cell cycle, how the cell can be moved from one phase to another, and how the cell can be moved from G1 to G0, and how can it be moved from G0 to G1. And I hope that I made things more clear to you. And next lecture I will be talking about mitosis and meiosis and checkpoints, the cell cycle checkpoints, and mitosis, meiosis, and spermatogenesis and oogenesis. Thank you so much for watching, and goodbye.